Hello, and welcome to my PH thesis defense of my project, The Selective Survival of E. coli in Freshwater Beach Sand. So for today's discussion, we'll start with a little bit of an introduction to myself and what motivated me to get into this field of research, as well as an introduction to my research question and why I'm interested in studying E. coli survival in beach sand. Next, we'll touch upon my project goals or objectives. What are the things that I wanted to determine through conducting this research? Next, we'll talk about the research methods that I used to conduct this work, as well as the results that we found and the conclusions that we were able to draw from this research. So first, a little bit about me. So I'm actually not from Wisconsin. I am a Pennsylvania native. And often I hear is, what does Pennsylvania have to do with freshwater sciences? Well, I'm actually from Erie, Pennsylvania, which is right here on the shores of Lake Erie. It's a great town if you ever have a chance to visit it. It's been going through a lot of changes in the past 20 years. Its economy used to be really based in manufacturing, but now it's really been shifting towards tourism. And one of the main drawers of tourists to this area is Presque Isle State Park, which is pictured right here, which is in my hometown. This is a beautiful state park that juts out into Lake Erie, and it's lined with a bunch of great freshwater beaches. Growing up in this area, I developed a great appreciation for the environment and for freshwater. So it really motivated me to study and to protect this area. So in 2016, I got my bachelor's in biology from Gannon University, where I graduated magna cum laude. While here, this is where I got my start in doing laboratory research, where I studied bacterial and chemical contamination in beach and drinking water. I really enjoyed working in the lab, so I decided that I should go for my PhD. So in 2016, I'd moved to Wisconsin and started at the University of Wisconsin-Milwaukee at the McLellan Lab in 2016. And here I've used molecular methods to assess bacterial populations. So what is the significance of E. coli and why is it used as a fecal indicator? So fecal pollution contains a lot of pathogens that can make us sick. And by fecal pollution, I do mean poop. So this is a complex substance that contains a lot of different pathogens and different viruses that can affect us. In fact, there's over 12 million cases of gastrointestinal illness and 50 million cases of acute respiratory diseases yearly worldwide due to exposure to contaminated recreational water. To be able to combat this, in the US, they passed the Clean Water and Beaches Act. And these acts mandate that recreational water is monitored for fecal pollution. So to be able to detect fecal pollution, it was decided that they should choose a single organism that's commonly found in it. Because it's a very complex matrix made of lots of different bacteria and viruses, they thought it would be good just to choose one to be able to use it as an indicator. And that if this fecal-associated organism is found in the environment, then it is assumed that it's from fecal pollution. And the organism that they decided to choose was E. coli. And there was a few reasons why they did this. So for one, E. coli is very common in the gut of warm-blooded organisms. This includes animals, humans, and birds. And it's found at a very high rate. So it is actually found, there's over 1 million E. coli per gram of fecal matter. Secondly, it was assumed that since E. coli is so host-associated, that it would decay quickly in the environment. That once it was passed outside of a host through fecal matter, that it would not be able to survive outside of the gut. And lastly, it was chosen because its enumeration or detection is fairly easy and cheap. So it's very accessible to lots of beaches, beach managers, and an easy method to use to be able to detect and determine how much E. coli is at a beach. However, the caveat is that current methods are non-specific, so it cannot tell an E. coli that is from a human from an E. coli that is from some sort of animal from one that is environmentally adapted. So overall, these were the reasons why E. coli is the most common fecal indicator used to assess for fecal pollution at beaches. So why am I interested in studying E. coli in the beach sand? So as I mentioned, E. coli is commonly associated with a primary environment, which is the gut of organisms. However, it's also been determined that E. coli is capable of long-term survival in secondary environments. And by secondary environments, I mean those that are outside of the gut of a host. So this can include in water, soil, or in this case, in beach sand. And beach sand seems to be a particular popular environment for E. coli to congregate. In fact, E. coli levels tend to be 10 to 100 times higher in beach sand than in paired water samples. And it was thought that these populations are forming here either through forming reservoirs, through beach water filtering through, or that there's even populations that are growing, propagating, and living here in this area. So there was great interest in understanding the origins of these populations and to be able to kind of assess these. So there's been some preliminary genetic analysis done, and what they found was that there's a subset of populations that are living in the sand that are genetically distinct from those E. coli that tend to be from hosts, such as humans or animals. So they did this using a few different genetic methods. So for one, they used DNA fingerprinting or multi-locus electroenzyme electrophoresis. So this just looks at different targets within genomes and then compares them, as well as multi-locus sequence typing, which is comparing the genetic sequencing of key genes between populations, as well as the use of DNA microarrays, which looks at the presence and absence of key genes in different populations to tell the differences. And overall, when they conducted this type of genetic analysis on E. coli populations from beach sand and compared it to populations from animals, they found that there were some genetically distinct populations living in the sand that were not found in any host, and that these appear to be populations that are living independently from hosts, so forming their own populations, propagating in the beach sand and replicating. So what's the significance of these non-host associated strains living in the beach sand? Well, these host-independent populations can be reintroduced into the water column. So when there's times of heavy rain or when there's times of high waves, these populations that are living in the sand can be reintroduced into the water. So then when they're sampling water, looking for E. coli that's assumed to be from fecal pollution, they're also capturing some of these E. coli populations that are not associated with the host. And these non-host associated strains that are found exclusively in the beach sand are not associated with the same health risks as those E. coli from fecal pollution. And these host-independent populations disrupt the correlation between the presence of E. coli and the presence of fecal pollution. So they reduce the accuracy of current beach monitoring methods that are aimed to detect fecal pollution. So as you can see here on this infograph, the E. coli that are able to survive in the beach sand have to withstand a lot of different selection pressures or stressors. They're exposed to UV light, changing in temperatures, changing in moisture and nutrient conditions. So those E. coli that can survive here must be able to put up with all these different stressors, and they must contain genetic factors or genes that allow them to be able to withstand these different changes and pressures in their environment. So we really wanted to be able to understand what are the genetic differences between these E. coli that can survive versus those that can't. And then once we find these genetic differences, we can use them to be able to detect these non-host populations and improve the accuracy of current water monitoring techniques. So what is exactly known about these environmentally hardy E. coli? So one thing to consider with E. coli is that there's a very high level of genetic diversity within these species. And this high level of genetic diversity allows it to occupy a lot of different niches or environments. Genetic diversity means that they can have a lot of different functions or factors that allow them to be different and to behave differently in these environments. So overall, there's several main phylotypes of E. coli, and these include A, B2, B1, C, D, E, and F, as well as some of these more rare cryptic clade strains, cryptic clades 1 through 5. And when you think of phylotypes or phylogeny, you can think of a family tree. So there's some phylotypes or groups that are more closely related, and there's others that are more genetically distant or not as closely related. And upon assessing populations from different hosts and from different environments, it was determined that certain phylogroups or certain branches of that family tree are associated with particular areas or particular environments. So for one, they found that B2 is generally more common with the gut of host, whereas phylogroups B1 and cryptic clades 2 through 5 tend to be more associated with secondary environments, and in particular in the beach sand. In fact, through an assessment of several freshwater beaches, it was determined that B1 was the most common phylotype recovered, so it makes up the majority of the populations that are found in the beach sand. As well as it was determined that cryptic clades 2 through 5 were found almost exclusively in secondary environments, so they were not found within the gut, but were rather found in the environment, in particular in the beach sand. Now, there's been some genetic assessment of what makes these cryptic clades able to survive in the environment. They did some whole genome comparison and found that these cryptic clades contain exclusive genes that allow for nutrient usage and competition. So, for example, it contains a pathway that allows for diol utilization, which is a type of nutrient that the other strains can't use, as well as genes for lysozyme production. This lysozyme production is an enzyme that degrades the cell wall surrounding bacteria, so it has a competitive edge in that open environment. And what we really wanted to determine is that are there other phylotypes that are capable of long-term survival in the beach sand? And are there other genes or genetic factors associated with these long-term survivors that can be used as an indicator of E. coli capable of survival in secondary environments? So as you can see here, this is the phylogenetic tree of E. coli over here. This side of the tree, you can see some samples from our main phylo groups, A, B1, E, B2, and you can see that they're somewhat genetically related, some more related than others, such as A and B1, which are sister groups, whereas B2 is a little bit more genetically distant. However, you have members of the cryptic clades over here, and you can see that their branching is longer, meaning that they are not as closely related as these other phylogroup members. So if you look up here on this circle part of the graph, the lines between each of these different samples represents the exchange of genetic material. So are they able to share different factors or functions? And as you can see up here, the cryptic clade members 2 through 5 readily exchange genetic material with each other, but they don't exchange genetic material with these main other phylogroups, indicating that they're forming independent populations that aren't trading genetic material. So they're forming their own populations with their own sets of genes that are not shared among these main groups. So we really wanted to expand this type of analysis and compare the survivability of different phylogroups and see if they contain their own sets of genes that are associated with survival. So what are the gaps in the research that were fulfilled by this work? So what did the research community need to gain? So previous work on E. coli survival in the beach sand has been pretty limited. It's focused on individual isolates from select phylogroups and sources. So instead of looking at E. coli in populations, how they naturally are found in the environment, a lot of the work is only focused on looking at specific strains or looking at E. coli from just particular sources. Secondly, the association of certain phylotypes of survival has been based on observation. Now, they take samples from the beach, they find high levels of B1, so they assume that B1 is better at survival than the other phylogroups, but it hasn't actually been demonstrated that this group is better at survival. And lastly, the genetic analysis of environmental survival has largely focused on cryptic clades. It's really been unknown if there are members of other phylogroups that are capable of long-term survival, or what kind of genetic factors that allow certain other phylogroups to be able to survive in beach sand. So overall, this work offers a much more robust picture of survival. For one thing, we focused on the survival of populations from multiple sources. So we took populations of E. coli isolated from sand, from gall waste, and from sewage, and then compared how these populations survived in the beach. So instead of looking at just a single phylogroup, we looked at how populations would survive, and this really mimics fecal deposition at the beach. So it was a good model to be able to demonstrate what E. coli out of a sample that are able to survive. Secondly, we wanted to be able to demonstrate experimentally that some phylotypes are better at survival than others. And we did this through our microcosm chamber, where we took microcosms or growth chambers, put populations in them, buried them at the beach, and then showed that some phylogroups survived better than others. And lastly, this work offered the most comprehensive genetic assessment of survival to date. We developed a novel population-level approach to identify genetic factors that are associated with survival. And this method can be applied beyond just this species or this environment. This is really a method that can be used to study any species in any environment to look at shifts and population changes throughout time. So overall for my project, I had three main thesis objectives or goals. So the first was to determine the survival and growth dynamics of E. coli in freshwater beach sand. And to do this, we use something called microcosms, or what you can think of as growth chambers. So first, we wanted to determine what the factors were that were limiting growth and survival in beach sand. So subjecting populations of E. coli to different treatments and seeing how that affected their survival. Secondly, if we wanted to compare the survival of populations from different sources, and these included sand, sewage, and gall waste. And as previously mentioned, this mimicked fecal deposition at the beach in a realistic way. So we looked at populations from sewage and gall that were active contributors of fecal contamination at the beach. So being able to understand how these different populations survive is really significant to understanding E. coli survival. Secondly, we wanted to determine if E. coli is selecting for specific phylotypes of E. coli. So in other words, we wanted to determine if there were members of a specific branch of the family tree that were better at survival than others. So to do this, we used something called Claremont phylotyping, and we did this with our microcosm survivors and those that were part of the initial population. So we took these populations of E. coli, put them at the beach, and then we assessed who survived. So looking at that initial population and the frequency of the different phylotypes and comparing that to those that survived, we were able to determine if some were better at survival than others. Secondly, we wanted to assess the distribution of phylotypes in beach sand and then compare them to the populations of sewage and gall populations. So we wanted to see if these sand populations looked unique from those from gall and sewage. And lastly, we wanted to define the accessory genome of E. coli capable of long-term survival, and we did this by using a novel population-level approach. So what we wanted to do was to be able to determine if there were any genetic factors common among those E. coli capable of survival. So to do this, we identified genes that increased in frequency by the end of the microcosm experiment. So looked for genes that were common among long-term environmental survivors. Secondly, we wanted to determine essential functional pathways that allowed for environmental survival. So once we identified genes that increased in the surviving populations, we wanted to determine what kind of function they played and how they worked together to allow for survival. And then lastly, we wanted to determine if any of these survival-associated genes were associated with any particular phylogroup. So for instance, does B1 contain any specific functions that these members of other phylogroups don't? So first we'll focus on the first thesis objective, determining the survival and growth dynamics of E. coli in freshwater beach sand. So to do this, as I mentioned, we used microcosm experiments, or you can think of these as growth chambers. So what we did was we took beach sand and then took our populations of E. coli and mixed them up in this beach sand and then filled up these little growth chambers with our beach sand mixed with the E. coli. We then sealed these up and then took them out to the beach, put them in this little cage, and then buried them. We let them incubate in the environment for 6 to 8 weeks, and then after 6 to 8 weeks, we came back, dug them back up, and then took our microcosms and then took them back to the lab. And at that point, we took the sand out of those microcosms and then re-isolated the E. coli out of them to be able to assess how these populations shifted throughout time. So a little bit more about our microcosm experiment design. So our microcosms were constructed of pieces of PVC pipe, and these pieces of PVC pipe had little end caps on them. On these end caps, we drilled a bunch of little holes and then lined them with 2.22 micron filters. And so what this allowed us to do was to be able to put our E. coli populations mixed in sand in them, with it allowing to still get moisture and air to be able to run through the system while keeping the E. coli that we were interested in that little chamber, while keeping the other bacteria from the environment out. And this allowed us to be able to study the survival of E. coli populations under real environmental conditions at the beach, opposed to other previous work, which largely focused on looking at E. coli survival under laboratory conditions. We felt that this design really mimicked what's really happening in the environment and would give us a realistic picture of E. coli survival. So to do this work, we conducted two different microcosm experiments. So the first was the nutrient competition microcosm. So we took the same population of E. coli that we isolated from beach sand. Excuse me. So we had about 188 strains of E. coli, and then we took these strains and then put them into 3 different treatments to compare how they were able to survive. So first we put these populations into native sand. So this was sand that was collected from the beach that was unaltered. So it contained that native microbial community that acted as competition, as well as contained available nutrients. So this is the amount of nutrients that those E. coli would typically be subjected to when they're deposited at the beach. Secondly, we put this population of E. coli into autoclave sand. So this was sand that was sterilized using autoclave. And how this works is that it uses pressure to lyse cells within the matrix, and those cells burst open. So what this does is this kills the competition, but the bursting of those cells releases all the nutrients that were within them. So this treatment represents no competition and high nutrient availability. Lastly, we subjected this population of E. coli to baked sand. So this was sand that was baked in an ultra-hot muffle furnace. So this was baked at 400 degrees Celsius for over 6 hours. And what this does is this kills the native population within that sand, as well as bakes off a lot of those released nutrients. So this treatment represented no competition and low nutrient availability. So comparing how this population survived in these different treatments gave us an idea of the different factors that might be contributing to E. coli survival at the beach. So next we did our source microcosm experiment. So in this experiment, we took populations from 3 different sources and then compared how they survived in the beach sand. And again, we had about 188 isolates or strains from each one of these sources. So we collected E. coli from beach sand, gall waste, and sewage. And as mentioned before, gall waste and sewage samples represent the deposition of fecal matter at the beach. So being able to see in real time how real E. coli fecal pollution is able to survive, and if there are any long-term survivors in this type of contamination. And when we did this, we took our little microcosm cage and we buried them about 1/2 meter deep in the berm sand for 6 to 8 weeks. So here on this infographic, you can see here is the berm sand, and this can be defined as the area of the beach that gets intermittent moisture. So you can think of this as when you go to the beach and you get there, the sand's really hot and you're burning your feet, that's the back shore, and then you are running to get to the berm, so that's that nice moist area of the sand. And this is where E. coli populations are the highest at the beach, which is in this berm. So that's why we decided to conduct our study right here in this area. So first, the results of our nutrient competition microcosm. So as mentioned previously, we took the same population of E. coli and then subjected them to different nutrient treatments to be able to compare how they survived. And this graph represents how those populations survived under these different treatments. And this is in log CFU per 100 grams of sand. So, and each marker, each dot here represents a different sampling time point. So you can see here in the yellow, we have our autoclave sample. So this is no competition and high nutrient availability. So you can see that when we subjected the population to this treatment, that it increased. So there was no competition in high available nutrients. So those E. coli were able to increase in prevalency. This is opposed to that bake treatment, which had no competition but low nutrient availability. So those populations declined over time, opposed to those in the native sand treatment, which had some available nutrients and some competition. So overall, you can see that those E. coli in the autoclave sand survive the best, and those in the baked sand survive the worst. Secondly, we also did what we called were nutrient spike-in trials. So what we did was we took some of our native sand that had the microbial competition and some of our baked sand that had zero competition and low to zero nutrients. And then we spiked each of these different types of sand with some of our autoclave sand that was high in nutrients. And when we did this, when we had our baked sand that had no other competition, and we put in a little bit of nutrients, you could see that those populations responded by increasing. Opposed to that native sand, where we added a little bit of extra nutrients, those populations didn't increase. This signifies that competition is affecting the ability for E. coli to survive. In the spike-in trial with the native sand, those E. coli that had competition were outcompeted for these nutrients, thus that population was not able to increase. So overall, through the results of the study, we determined that nutrient availability as well as competition appear to be affecting E. coli survival in beach sand. So next was our source microcosm experiment. So as mentioned before, this was the experiment where we took E. coli from 3 different sources and then compared how they were surviving in the beach sand. So you have our autoclave here in the dashed line, and our native sand treatments in the solid line. So again, autoclave sand, no competition and high nutrient availability, and you have our native sand here, which has competition and low nutrient availability. And we have our sand populations in the yellow, our autoclave, or our sewage populations in the red, and our gall populations in the green. So overall, you can see that E. coli from each source tested were capable of survival. So at the end of each experiment and each trial, we were able to recover surviving E. coli. And that there wasn't much difference in the ability for E. coli from these different sources to be able to survive. Secondly, we were able to determine that the autoclave sand treatments survive better than the native sand treatments. So again, demonstrating that nutrient availability and competition were affecting E. coli's ability to survive. So what we were really interested in looking at next was looking further at these native sand populations, as this treatment most mimics what's occurring at the beach, as we used sand that we collected from the beach that was unaltered and then subjected our populations of E. coli to them. So we wanted to determine how the populations from the input of the experiment differed from the end survivors. So do these surviving populations contain specific genes that allow them to survive? Were there a particular branch of the family tree that composed more of these survivors? And that leads me to my second thesis objective: determine if freshwater beach sand is selecting for specific phylotypes of E. coli. So to be able to do this, we employed something called Claremont phylotyping. And what Claremont phylotyping is a multiplex PCR. And what that means is that it allows you to detect the presence and absence of genes in an E. coli strain. And this presence and absence of genes is able to determine what phylotype that it belongs to. And it's able to determine if E. coli belongs to the following classifications: A, C or E, B1, B2, D or E, F or unknown, or clade or E. So as you can see, this top image is an example of a gel electrophoresis. So when you conduct this PCR, this endpoint PCR, to be able to visualize your results, you use this gel electrophoresis. And each lane represents a different E. coli strain or sample, and then the different bands in them indicate the presence or absence of that gene. If there's a band, that means that that sample contains that gene. So based on the presence and absence of these 4 different genes, you're able to determine which phylotype that E. coli sample belongs to. seen in this graphic down here. So these boxes represent each of the 4 different genes, and then based on the presence and absence of these genes, you're able to determine what E. coli phenotype or what branch of the family tree that sample belongs to. To be able to assess these populations to determine if they shifted in a meaningful way, we employed the chi-square goodness of fit statistics. And this allows you to determine if expected and observed distributions of nominal data are statistically different. In other words, did these populations shift in a meaningful way, or did the populations kind of stay the same? So first, we just wanted to compare the phenotypic distribution of our 3 different E. coli sources. So this is a graph in relative abundance, and it's color-coded by the different phylotypes. So you can see our gall population had a pretty even distribution of phylogroups. There really wasn't one particular phylogroup that was dominating it, opposed to the sand and the sewage populations, which were both dominated by this B1 phylotype. Interestingly, sand and sewage are considered secondary environments, and this matches what the literature tends to say, is that B1 is common in secondary environments. So our findings were consistent with the findings of the greater research community, opposed to the gall sample, which is a primary environment where it wasn't really predominated by a single phylogroup. So when we did our chi-square goodness of fit, comparing the distribution of these different populations, we found that our primary environment sample gall was different from the 2 secondary environments. So the gall population was significantly different from the sand population, and the gall population was significantly different from the sewage population. However, when we compared our 2 secondary environments, our sand and sewage populations, we found that they were not significantly different. So next we wanted to see how these populations shifted after incubation at the beach. So we took our populations, put them in those little microcosm or growth chambers, and then buried them at the beach for 45 days, dug them back up, and then re-isolated that E. coli, and then assessed the distribution of different phyllotypes in those populations. And as you can see, the surviving populations were largely predominated by members of phylogroup B1, as well as there was an increase in members of phylogroup A, which is interesting because A and B1 are sister phylotypes, so they're very closely related. And that those from phylogroup B2, which are generally more host-associated, generally decline in each of these populations as well. And when we did our chi-square goodness of fit, comparing our initial populations to our output populations, we found that in each of our populations tested that they were significantly different, meaning that these populations shifted throughout the experiment, that there was differences in the initial populations and the end populations. So we found experimentally that B1 was the most common phylogroup recovered, but we really wanted to be able to see if this was what we'd also see in the actual real world in the environment. So what we did was we collected beach sand from 4 different beaches. So 2 of these beaches were considered urban beaches, so these were Bradford Beach and Atwater Beach right in Milwaukee. And then we also collected sand samples from Coeur Andre Beach and Point Beach, which were up north in a more pristine or rural area. And then we did our Claremont multiplex reaction to be able to determine what the phylotype distribution at these different beaches were. So overall, you can see that often B1 is the most common phylotype recovered from beaches. Interestingly, there are some beaches where there is high levels of B2 or D or F or D or E, which are commonly more associated with the gut, and that these beaches that have more of these gut-associated strains are generally more urban beaches, so closer to the beach, closer to the city. So meaning that there might be different sources of E. coli coming from the area depending on if it's a rural or an urban beach. So we wanted to compare these urban beaches to the rural beaches. So when we combined our samples, we saw a similar pattern where B1 was the most predominant phylogroup, But we also found that in the urban beaches, there did tend to be more of these B2 or D or E's, which as mentioned are more human associated. We also wanted to determine if we would see the same pattern in the gall, that even distribution of phylogroups. So we recovered another 200 isolates from gall samples, and again we saw this same distribution of phylotypes. So our experimental results of B1 being a strong survivor are reflected in the environment where B1 is commonly recovered. So next, we really wanted to get into the genomics of survival and to determine what makes these long-term survivors genetically unique. And that brings us to our third thesis objective, defining the accessory genome of E. coli capable of long-term survival. And this was done using a novel bioinformatics approach. So first we'll start with an overview of my genomic analysis. So this square right here represents our population from our microcosm input. So those initial populations we put into our growth chamber. And it's color-coded by the ability for the E. coli to survive. So each oval represents a different E. coli strain, and then the color represents its ability to survive. So you can see here in the teal, we have some E. coli that are weak environmental survivors, and in the orange and the yellow, you have some that are intermediate survivors, and then you have the purple ones, and those are strains that are capable of strong environmental survival. So this is what the population looked like after incubation at the beach. So we put those populations in the growth chambers, buried them at the beach for 45 days, and then dug them back up. And after that 45 days under environmental stressors or pressures, you can see that those that contain genes that allow for strong environmental survivorship increased in frequency in the population, as the weak survivors and the intermediate survivors decreased. So these strong survivors made up more of this population. So to be able to assess these populations, we decided to do a novel thing with our genomic analysis. So typically, when you're looking at E. coli or looking at the genomics of different organisms, what you would do is you would genetically sequence each isolate or strain separately. So if we did this way, our sequencing budget would run out pretty quickly. We would only be able to sequence the genome of about 50 to 100 isolates. So we decided to take a unique approach where we took these populations of E. coli from each source and each time point and sequenced them as a whole. So for example, for our gall population, we had about 200 isolates in that initial population. So we took equal concentrations of each one of these strains, pulled them together, and then sequenced them as a whole. So we have the whole genomic contents of that initial gall population. And then we did that again with our output population. So after 45 days buried in the beach, we re-isolated those E. coli, and we took equal concentrations from each of those surviving isolates, pulled them together, and had the genetic composition of those surviving populations. So comparing the genomics of a whole of a populations from beginning and end of the experiment from each of our 3 different sources. So how are we able to analyze this pooled genetic material? How are we able to identify genes that were more common among survivors? So to do that, we used something called mapping. So what we did was we took accessory genes from E. coli, and then we used that as a scaffold. And we took our genomic sequences, our short-read genomic sequences, and then mapped them to this scaffold. So you can kind of think of this as a jigsaw puzzle. So each sequence of DNA you can think of as a puzzle piece, and if it's matching to that scaffold, it will map to it. And then the more copies of that gene that are within that sample, the more puzzle pieces are going to fit in that area, so the higher depth of coverage. So for example here, let's pretend that this dark line is an accessory gene that's associated with long-term survivors. You can see that in this initial population, there were some copies of this gene, so there were some long-term survivors. However, at the end of the population, in our output population, there was much more of these long-term survivors containing these long-term survivor genes, meaning that there are more of these genomic sequences, and that this resulted in a higher depth of coverage of these accessory genes. So more copies of the genes, more prevalent in the population. So comparing the mapping coverages of accessory genes, we were able to identify genes that were more common in our output populations, while genes that were increased in frequency and were found more prevalently among survivors. So once we used this method to identify genes that were more common among output survivors, we ran it through a program called Ghost Koala. And this program allows you to identify the functions of genes. So it allows you to determine what genes are there, and not only what genes are there, but how they interact to produce different factors or form different functional pathways. So for example, we looked at functional pathway modules. So these are groups of genes functioning together to carry out some sort of action. So in this case, we found that long-term survivors contain a pathway for betaine biosynthesis, which is a powerful osmoprotectant and allows E. coli to be able to not dry out when they're under moisture stress. So if you look here, this outer black box represents the module, and then each tiny red box within it represents a different gene. You can see that in our output population, we contained each of the genes in this pathway, meaning that this pathway was enriched in surviving populations. So it was common among survivors. So indicating that this pathway aided in environmental survival. So the first step in doing this analysis was identifying genes to use in our scaffold. So those genes that were used to map our short read sequences from our composite populations to. And to do this, we did a pangenomic analysis. And what it means is that you compare the genomic content of different strains and identify which ones are found in all of them and which are variable among the population. So overall, pangenome means it's all the genes within a population. And it consists of two parts. So the core genome, which are genes shared by all members of that species or population, and the accessory genome, which are genes that aren't found in all members. It's variable amongst the different strains. So as you can see here on our graph, this is our pangenome, and as mentioned, this assesses the presence and absence of genes in a set of genomes. So each different outer ring represents a different genome, and this is color-coded by phylotype. And if there's a shaded area in that genome, that means that that gene is present. So each shaded region represents a different gene. And if it's shaded in that sample, that means that that sample contains it. So you can see here where there's all this shaded region, you can see all these different isolate strains contain this gene. So these were considered our core genes. But we were really interested in looking at these accessory genes that varied amongst populations. So what we did was we got rid of our core genome and then just focused on these accessory genes. And as mentioned, we took these accessory genes and then used them to build our scaffold. So we took each one of these genes and concatenated them or lined them up back to back and then mapped our short read samples to them. So this is an example of our microcosm mapping results. So as mentioned, we took our accessory genes and made the scaffold with it and then mapped these short read samples to it. So on this graph, if you imagine the inner circle here is our accessory genes. So all those different genes that were variable amongst our population. And then each outer ring of here represents one of our different composite samples from each time point and source. And these were all done in triplicate. You can see our input populations are in our darker colors, and our output population, or those that survived the incubation in the sand, were in our brighter colors. So each ring represents a different sample, and then with each ring you see this shading. And the depth of shading in this graph represents the amount of coverage or the amount of short reads that were mapped to that gene. So around the inner ring our accessory genes, and then around these outer rings, the depth of coverage or the amount of short reads that matched. So what we did was we compared our input populations and our output populations among each source. And then looking at that different depth of coverage or amount of shading, we were able to determine those E. coli that were, or those genes that were more common among our E. coli surviving populations. So we used a cutoff of genes that were enriched greater than 25% in our output populations as those that were associated with survival. So as you can see over here, for example, you can see in this dark green, this represents our initial gall population from the beginning of the experiment. And then we have our bright green samples, and this represents the gall populations that survived the experiment. And you can see this particular set of genes, there's a higher depth of coverage in the output populations than the input populations, signifying that this gene increased in frequency throughout the experiment. So that genes that contained E. coli that contained this gene were able to survive and increase in frequency. So genes that were more common among surviving populations than initial populations. So we did this with each one of our accessory genes and identified those genes that were more common among surviving populations. So we took these surviving associated genes and then assessed them through Ghost Koala. So first we looked at the distribution of these enriched genes among survivors. So overall, we identified a total of 440 unique functions that were enriched by at least 25% in survivors. The SAN populations contained the highest number of these functions, so there was 356 of these surviving populations in the SAN. There were 326 of these functions found in the gall populations and 306 found in sewage. So when we compared the distribution of these different functions among our groups through a Venn diagram analysis, you can see that the sand and the sewage populations and the sand and the gall populations shared a significant number of unique functions, opposed to the sewage and the gall populations, which didn't share many of these functions. To me, this signified that the sand is receiving influence from both sewage and gall, so that there's populations being deposited here from each of these sources, and that this is reflected by having common genes with these sand populations. Whereas the gall and the sewage, they don't really share populations among them, so they share a low number of functions. So overall, we were most interested in functions that were found in the survivors from each of these different sources. So we wanted to find functions that were common among sand survivors, among sewage populations, and among gall surviving populations. And overall, we found 198 functions that were shared among the survivors from each of these populations. So we took these 198 functions and then analyzed them with bright analysis and a module pathway analysis to assess the functional relationship among them. So first, we'll talk a little bit about our bright analysis. So bright analysis is the hierarchical classification of functions. And what that means is that it allows you to take a gene and determine what protein family it belongs to, and then what kind of function it plays within that family. So as you can see, the majority of these shared enriched functions were found to function in metabolism, and specifically metabolism enzymes, as well as signaling and cellular processes, in particular functioning as transporters. And these both largely had to do with the breakdown and transport of carbohydrate and amino acids, two of the main most common and important nutrient sources for organisms. So together, these functions work together to form functional units called modules. So as shown earlier, here's an example of a pathway module. And when we ran this type of analysis to see how these different genes function together, we uncovered several pathways that were common among survivors. So the first was for biotin biosynthesis, as previously mentioned. This is a pathway that forms a molecule that acts as a powerful osmoprotectant. So this prevents E. coli from being desiccated. It's able to help those that contain this function to be able to withstand different moisture conditions. Secondly, in surviving populations, we found pathways for GABA biosynthesis in the GABA shunt, and these pathways are very important because they allow for nutrient use flexibility as well as pH balance. So again, allowing the owners of these functions to be able to adapt to the changing environments in the beach, such as nutrient availability as well as changing chemical conditions such as changing pH. And then lastly, in the surviving populations of the sand and gall populations, we found a pathway for histidine degradation. So signifying that there may be some unique long-term survivors found in gall that were not found in sewage. So next we wanted to do a phylotypic assessment of these enriched functions. So we found these enriched functions and we wanted to determine if they were associated with any particular phylogroup. So overall, of the 198 functions found in each of our surviving populations, the majority were not associated with a specific phylotype. They were generally associated with a subset of members from each of the different phylogroups. However, we did find 16 functions that were associated only with members of A, B1, and or the cryptic clades. So these functions were not found in members of other phylogroups. And these largely had to do with transport and metabolism enzyme. And interestingly, these functions were shared among A, B, and the cryptic clades, which is very interesting because A and B, as you can see, are sister clades, so they're very closely related, so expected to exchange genetic material, share similar functions. However, these cryptic clade isolates are genetically distinct and are not thought to exchange genetic material with these other groups. This is why it's thought that these 16 functions could be representing a E. coli beach sand ecotype. So this means that in the environment, the different pressures select for different strains that contain these functions that weren't necessarily evolved together, but evolved separately in these different groups. So evolved differently in the cryptic clades versus through A and B1. And lastly, it was determined that not all members of these strong surviving groups were able to survive in the environment. So when we did our pangenome analysis, we found some A and B1 exclusive genes that were not enriched in survivors. So there were some strains of A and B1 that didn't increase in frequency in the experiment. So meaning that only certain members of these phylogroups survived, and that these members are most likely those that contain these special unique functions. So next we wanted to conduct a phylotypic association of our enriched modules. So we found these functional pathways enriched in survivors. So we wanted to see if there were any phylotypic assessment or association with these survivors. So overall, you can see our main enriched pathways for betaine biosynthesis, GABA biosynthesis, and the GABA shunt were widely distributed amongst most of the main phylogroup members, so common among A phylogroup members, B1, D, and F. However, when you look at their distribution in cryptic clade members or B2 members, they're variable. So for instance, looking at these B2 members, you can see that they generally do not contain the pathway for GABA biosynthesis. So perhaps this represents a split in the family tree. So you can see B2 is kind of genetically distant from some of these other members. So the ownership of this pathway may be, a difference between these different groups. And then looking at these cryptic clade members, you can see that they generally, the ownership of these survival-associated functions varied amongst them. So this suggests to me that the cryptic clade members might take different strategies to allow them to survive in the environment, opposed to members of the main phylogroups, which seem to readily contain these survival-associated functions. So again, representing a split in the genetic tree, where cryptic clades might contain their own set of functions because they don't share genetic material with these other groups, where these other groups that share genetic material contain and share these pathways. So overall, with this work, I was able to come up with three main conclusions. So the first was that the survival of E. coli in beach sand is affected by competition and nutrient availability. So we determined this through looking at our microcosm experiment. So overall, there were higher levels of survival in the autoclave sand and nutrient spike-in trials. So those populations that had low competition and high nutrient availability survived better, indicating that these are factors that affect E. coli survival in beach sand. Secondly, we were able to determine that E. coli survival is much more widespread than expected. It was originally thought that E. coli was unable to survive in the environment, but we proved that E. coli from many different sources were capable of long-term survival. And we determined this as a subset of E. coli from each source and phylotype were able to survive. Secondly, we determined that the majority of survival-enriched functions were widely distributed among phylotypes, so meaning that members from different phylogroups are capable of some level of environmental survival. And then lastly, we found that the survival-enriched functions largely aided in nutrient use flexibility and the adaptation to changing environments. So very key important factors in E. coli survival in the beach sand. And lastly, we were able to determine that though a subset of all phylogroups were able to survive, A and B1 members did appear to perform better in the environment. And this was determined as seeing that members of A and B1 increased in frequency throughout the microcosm experiment. So they were more common among our surviving populations, as well as when we did our beach survey, we found that B1 predominated many of our beach samples collected. So what is the significance of these findings? How do we apply these findings to the real world to be able to improve beach monitoring? So for one, we provided evidence that E. coli is capable of long-term survival in beach sand. This really disrupts the correlation between the presence of E. coli and fecal pollution. So proving that current beach monitoring methods need to be improved and that just looking at E. coli populations simply isn't good enough. Secondly, we demonstrated that nutrient concentration increases E. coli levels in the sand. This suggests that if we employ nutrient mitigations at beaches, such as rain gardens, which can capture nutrients from runoff, can improve beach hygiene and thus improve the use of E. coli as a monitoring tool. So if you're able to reduce the amounts of nutrients that are entering into the beach sand, this will reduce the amount of these sand E. coli populations and thus reduce the amount that enters into the water. Thirdly, we were able to identify potential targets that can be used to develop molecular tools that can be used to identify these long-term survivors. So using some of these pathways that were only found in long-term survivors, using these genes to be able to differentiate long-term survivors from those from recent fecal pollution. And this can be used to improve the interpretation of beach monitoring results. So if you have a chronically contaminated beach, you can look and see if those E. coli that are there are more long-term survivors or if it's a mix of those from fecal pollution. And then lastly, we provided a novel method that was used to assess level populations at a new way. So using population level sequencing opposed to looking at just comparing the genomic analysis of single isolates. And this allows for a robust picture of how populations shift under different conditions. And this offers the research community a new and efficient tool to assess populations. So you get more bang for your buck for your genetic sequencing budget, and you're able to assess more holistically how populations shift and change. So I'd like to take a moment just to thank you all for listening today. Thank you all along the journey, especially for my supervisor, Dr. Sandra McClellan, all the postdocs and lab mates I've had and undergraduates I've helped with, as well as my wonderful high school volunteer. So thank you.